Welcome to section 9.4, where we will talk about skeletal muscle fiber physiology. So we know that our nervous system is going to help control our muscle contractions through their action potentials. So just like how our nerve cell had a resting membrane potential, our muscle cell is also going to have a resting membrane potential. And that's going to be the membrane voltage difference across the membranes. They are polarized, meaning the inside of our cell is more negative because we've got a large amount of negatively charged proteins. And we've also got more potassium on the inside than our outside. The extracellular fluid is going to contain more sodium ions, and we will have some potassium that'll leak out, but not completely because we also have open leakage channels that will allow it to move back in to its negative proteins. And just like in our nerve cell, we will also have a sodium-potassium pump that will help to maintain this polarity. And we have to have this setup of polarity in order for an action potential to be able to occur and ultimately cause a contraction to take place. So let's review some of the types of ion channels that we have. Ligand-gated channels are really important when we discuss the neuromuscular junction. Do you remember that? We will have a channel which has specific receptor sites that will bind a neurotransmitter. So in this case, for muscle, it'll be acetylcholine will bind to this ligand-gated channel. And when it does bind, that will cause that channel to open. And when it opens, sodium will flow through and cause the inside of our muscle to become more positive. Now for our voltage-gated channels, they will always remain closed until we achieve a specific level of voltage. When we reach that specific level of voltage, called the threshold, at that point the voltage-gated channel will open up and we get more sodium to flow through, which will cause the inside of our muscle cell to become more positive. Now, it doesn't have to be sodium all of the time, but in this instance, when we are talking about our skeletal muscle cells, that's what it is. So with our resting membrane potential in our resting cell, we have our purple ions here that are representing potassium, and we have more of those inside the cell. We've got our green negative proteins here. And our pink ions are representing sodium, and we see more of those extracellularly. Notice how there are more potassium leakage channels than there are sodium leakage channels. And you could see that each of these ions, whether it is potassium or sodium, they're always going to flow in the direction of their concentration gradient. So we have a lot of potassium here and not as much potassium out here. So potassium is typically going to want to flow from inside the cell out, because it wants to go to have more space. There isn't a lot of potassium out here, so it goes to that region. Whereas sodium, we've got a lot of sodium out here and not so much inside our cell. So sodium is naturally going to want to flow through its concentration gradient and move from outside the cell into the cell. But because the inside of our cell here is so negative, when potassium flows out, it wants to flow back in because it's being pulled by its electrical charge back inside the cell. Whereas sodium doesn't really have that electrical charge out here. It wants to stay in here. So it'll always be drawn inside the cell whenever possible. And we also have our sodium-potassium pump that will help to maintain this different ion concentration of more potassium inside the cell and more sodium outside of the cell by pumping 3 sodiums out and 2 potassiums in. Now let's review some of these terms that we've already discussed in the nervous system. We've got depolarization of our plasma membrane, and that's going to cause us to become less negative. So we are moving positively up in this graph. If we reach this threshold, which is going to be our end plate potential, causing enough change in the membrane voltage to reach this critical level, then we will get our full depolarization, which is now called an action potential. When we reach the top of our action potential at positive 20 here, then we are going to close those sodium doors of the voltage gated channels and open the potassium voltage gated channels, allowing potassium to flow out of the cell, and that helps to return the resting membrane potential back. And sometimes we can go a little bit lower than what the resting membrane potential is, and at that point it would be called hyperpolarization. But eventually the sodium and potassium pump would restore that resting membrane potential. We are also going to have that all or none principle take place here. So like a camera flash system, all action potentials are going to be identical. It either happens or it doesn't. We'll also use this term called propagate or travel as I describe how we transmit the action potential from the nerve to the muscle cell in order to get it to contract. So basically the action potential is spreading from one location to another, so that the action potential is going to travel and create a new action potential at each successive location. And frequency will be the number of action potentials that are produced per unit of time. So for a stronger stimulus, we need to increase the frequency of the action potentials that take place. So this illustration will show you what takes place with our gated ion channels when we have an action potential take place. Here we've got just our leakage channel open, lots of potassium inside, lots of sodium outside, and this is our cell at rest. When we have depolarization take place, the ion channels will open up and allow sodium to move in. So we get an influx of sodium. When that happens, we get depolarization that is taking place, and if we hit that threshold, we get the action potential. So once we hit our positive 20, positive 30, whatever it is, the end of our action potential, then we get repolarization that is going to take place. Our sodium ion channels will close and the potassium ion channels will open, and all the sodium inside the cell will want to flow out, and that will cause the inside of our cell to become more negative and get back to its resting membrane potential. So here's action potential propagation. This is now our muscle fiber, and we have an action potential taking place here. And as we're getting all this, let's say, sodium flowing in, causing the inside of the cell to become positive, some of that sodium flows over to this area and will cause an action potential to take place in that region now. As this area is going through repolarization and getting back to its resting membrane potential, we will get that same thing happen all over again. Some of that sodium moved over to the side of our muscle fiber, and an action potential is now taking place over here. So once we get that action potential to travel through that nerve and get to the axon terminal, we are going to have these vesicles filled with acetylcholine move close to the presynaptic membrane and leave that neuron through exocytosis. The acetylcholine will then travel over to these ligand-gated receptors, attach to the ligand-gated receptor, and cause it to open. And so now sodium is going to flow into the muscle cell and cause that action potential to continue along this sarcolemma. We will also have acetylcholinesterase present to help to degrade acetylcholine. So once it's done attaching to the ligand-gated receptor, acetylcholine will be released and acetylcholinesterase will break it apart into acetic acid and choline. And this prevents the accumulation of acetylcholine so that that muscle can then relax or go back to its resting state. So here's the full scheme of how this takes place. We get the action potential to move toward the axon terminal, and when we have these calcium voltage gated channels sense that voltage change coming from the action potential, it takes calcium from the extracellular space and moves it into the axon terminal. Calcium will then go and bind to our synaptic vesicle, causing it to leave the neuron through exocytosis. Acetylcholine will then attach to our ligand-gated channel, which causes it to open and sodium to move into our muscle cell. Acetylcholine will then unbind from the ligand-gated channel, and when it leaves, the channel will close, so no more sodium can move in. We will have acetylcholinesterase then break apart acetylcholine. Acetic acid will kind of diffuse away from the synaptic cleft, and choline will be recycled and moved back into the neuron so it could be reused to make more acetylcholines and store them back in the synaptic vesicles for future use. Next, let's talk about excitation, contraction, coupling. This is going to help to link our electrical and mechanical components of contraction, meaning it takes that action potential and causes mechanical movements, let's say, in order for the muscle to contract. The action potential that was sent from the neuron over to the sarcolemma of our muscle will then propagate into our T-tubules. So let's say we have a neuron that would be over here. That action potential is propagating along the sarcolemma and reaches our T-tubule. It will then grab calcium from our SR, which is in yellow here, the Sarcoplasmic Reticulum, and that calcium enters the sarcoplasm, which would be in here, and binds to the troponin associated with the actin myofilament, shifts that tropomyosin we talked about, and the myosin head attaches to the actin and causes the muscle to contract. So let's get a little bit better visual as to how this all takes place. We got that action potential propagating across the sarcolemma. It's traveling down the T-tubule, and you'll see in an upcoming slide that there are calcium-gated channels in the SR here in order to open and allow the calcium to move out and reach the troponin, which is in red here. That troponin will shift the tropomyosin that we see over here, the rope-like structure, that will reveal to us the active sites. And the myosin head will attach to the active site and cause the power stroke in order for the contraction to take place. This slide is just to give you a little bit more detail as to what's taking place in our triad. Triad meaning our T-tubule and the two cisterna of our SR, the Sarcoplasmic Reticulum. So it's 1, 2, 3 structures at play here. You don't have to memorize this slide. I just think it really helps to conceptually know what's taking place so it all kind of makes sense. So what we see here is our T-tubule, and this T-tubule is going to have an integral membrane protein called the DHP receptor that will protrude into the intermembrane space, which is the space between the tubule and the muscle fiber sarcolemma. Then we have our SR cisterna membranes that are going to be associated with integral membrane proteins as well. But this one is called the ryanodine receptors that will also protrude into the intermembrane space. These proteins or receptors are going to help to control the opening of the calcium channels in the SR cisterns. So if we look at one that doesn't have an action potential stimulating it, it stays closed. So the calcium is going to stay within the SR. But if we get an action potential that comes through, so let's say it propagates down the sarcolemma and down the T-tubule, then we get that signal that's transmitted over to this voltage-gated calcium channel, and it is going to open and allow the calcium to leave the SR. So imagine the calcium is in here. This calcium channel opens up and allows the calcium to move down and toward the cytoplasm, where we're going to find our troponin that is associated with the actin myofilament. That binding of the calcium to troponin will then shift the tropomyosin out of the way, and those active sites will have a myosin head attached to it, causing the power stroke and the contraction taking place. So now let's take a look at this cross bridge movement that is taking place. So as we said before, calcium moved down attached to the troponin, shifting the rope-like tropomyosin out of the way. We see the active sites now revealed on our actin myofilament. That myosin head is now able to attach to that active site, and when it does, phosphate is going to leave it, leaving behind ADP. And when that phosphate leaves, we are now able to create a power stroke. So the energy that was stored in the myosin head from phosphate leaving ADP causes the shift of the actin myofilament to slide past the myosin myofilament, and then the ADP is released from the myosin head. Then we have to have another ATP molecule come over and attach to the myosin head in order to release the head from the actin myofilament. If ATP doesn't attach to myosin, then that myosin head stays attached to the active site. ATP is then going to hydrolyze, breaking into its inorganic phosphate and ADP, and those will stay attached to the myosin head. The last phase of this is the recovery stroke. So the head of the myosin can kind of go back to its resting position, and energy will be stored in the head so that it can reattach to the active site if calcium is still bound to troponin. So the cycle will take place over and over again during a muscle contraction, because it's not just one power stroke that's needed. It bounces up and attaches and does its power stroke a couple of times in order to get the actin filament to move all the way toward the H zone. If it does it just once, it's not going to be enough. Now let's do a full summary of skeletal muscle contraction. We've got action potential moving down our motor neuron, causes the voltage-gated calcium channels to open, allowing calcium to move in, bind to our synaptic vesicles filled with acetylcholine, causing it to exocytose. That acetylcholine then attaches to our ligand-gated channel, causing it to open, and sodium to move inside our muscle cell. That action potential is now going to propagate down the sarcolemma until it heads over to our T-tubules, causing calcium-gated channels here to open, and calcium will move down. So we're going to switch to this side, I guess. Calcium is going to move down to the cytoplasm where we will find troponin. So we're going to go to this picture here. Calcium binds to troponin. That shifts tropomyosin out of the way, revealing the active sites, and myosin head can now attach to that active site. And when it releases that inorganic phosphate, the power stroke takes place and the actin filament will move toward the H zone or the M line. So that actin myosin on this side will move in this way, and the actin myofilaments on this side will move in this way. Now, how does muscle relaxation take place? When we have no more acetylcholine in our neuromuscular junction, that calcium is going to move back into the SR by active transport, meaning we require energy in order to do this. Calcium will move away from that troponin-tropomyosin complex. That means tropomyosin will move back to its original position and cover up those active sites. So now the myosin heads can't bind to the active site in order to cause actin to move in, causing muscle contraction. So that is it for section 9.4. Stay tuned for section 9.5.